Last week, or before, we talked about 3D comparisons, and last week we began the 1D comparison story, and today we will continue on the 1D comparison. Let me have a short recap. So there is the notion of 1D, 2D, 3D. 3D structure is essentially the coordinates of the structure, 2D are the contexts, and 1D is a string. We'll get back to that next week. There are 3D comparisons give you families of related proteins, as shown here. So you have 50 different structures that somehow are very, very similar. So that's why they superpose so well that this looks almost like one single structure where you have the helices and the strands. Dynamic programming is essentially a brute force simplified by the fact that you have a limited number of steps you can go through the matrix, and it achieves an optimal superposition of two sequences, again, sequence-wise alignments, optimal by some objective function, not necessarily by the biological objective. We talked about the fast matching, BLAST, and the fast matching starts with a hashing, with essentially word matches, where a word match is not defined by identity of letters, but it's defined by similarity higher than something in a scoring matrix. The important issue here, so then the initial word matches are extended in both directions. And the important issue here really is to sort out the background statistics, meaning that you need to figure out what's the probability to get a certain match randomly, by chance. I began talking about what I tend to refer to as the zones. So when two proteins are very sequence similar, sequence alignment is trivial. And then we get into some region here where the alignment gets very tricky, Twilight zone. And then in a region where the alignment gets impossible. And this transition I'd like to reflect by numbers. And the numbers could essentially be like this one here. It could be that, so if the Twilight zone started here, or this would still be the safe zone described as we don't make any mistakes. Red are the mistakes you make. So this is percentage sequence identity. That's the percentage or the number you find. And essentially it means the less similar two sequences are, the more you will find incorrect ones, red. Correct, incorrect, blue and red here is defined as proteins that have the same structure versus in red proteins that do not have the same structure. So blue proteins, pairs of proteins of similar structure, and red proteins of different structure. So the first question today for you, I'd like you to sort of come up with a little grouping and answers, is how could you possibly, this slide before here was a hypothetical slide. We expect something like that. Those are not real data. How could you create a background set to measure this? To see what the real curve, the real red, the real blue would be like. How could you do that? Anybody has an idea? So again, what you want to figure out, so you align two sequences, you use, for instance, dynamic programming. So the alignment itself is not the problem, but you're asking how well does the alignment reflect the biological reality? The biological reality is you want to say that two proteins that have similar structure are related in structure, and you want to say that proteins that have different structures are not related in structure. So you want to sort of sort your pairs into two bins. The alignment is a given. The sequence alignment is, so you start with a bunch of sequences. My question is where you get the database or the background or the standard of truth. Any ideas? Could you speak up a little bit?
Previous experiments, do we have?
So we have previous experiments. What do you do with previous experiments?
We can compare it with them if we have a standard.
So what exactly do you, so say I would be your dream experimentalist. I give you whatever you want. What do you want? Or somebody else? So again, the question is, those curves are fake, or are imagined, or hypothetical. How could we make them real? What we want to see is when the identity in the letter between two proteins drops, from right to left it drops, how many false positives, how many true positives do I find? What is this safe zone, to go back into this image here? What is this zone in which I clearly identify that two proteins have a similar structure? How can I do that? I can use experimental information. Can somebody suspect how? What type of experimental information do you want from me in order to design this? Again, the goal is to figure out at what level of sequence identity do two proteins have a similar structure. What do you start with? Is that too trivial, or? So if I made person X say it, would person X know that? Or is it too complicated? So the shaking of the head means that it's too complicated, or you don't know, or I hear my victim because the only one who moves. Everybody is just frozen. Yeah?
Could you maybe explain one more time the zones?
So, great, thank you. So what I mean here is, again, the dynamic programming principle gives us an optimal alignment. We have two sequences, we superpose them, and I say optimal. But optimal is defined by what is called an objective function. One objective function could be match the identity of letters. One simple objective function. Now that objective function is not necessarily relevant for biology. In biology, and here is the BLAST, is a similar idea. Now, what is relevant for biology in this particular case, for this particular lecture here really, is that I want to match structures. So I want to say from the alignment to this pair of sequences, so I have only the sequence, I align them, but I can say from the alignment that they have similar structure. When those two have 100% sequence identity, that's trivial. When they're very, 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 very similar, I told you similar sequences have similar structure, so this is very likely. At some point here, the signal will drop. At some point, I will pick up things, because the sequence identity will be so low, I will pick up things that no longer have the same 3D structure. Safe zone here is the one where I safely can infer without any mistake, essentially, that's the idea, that I don't make a mistake in inferring that two proteins have a similar structure. Again, I start with the sequence alignment. I don't know anything about their structure. I align the sequences, and somewhere here, and how exactly this zone extends here, we want to figure out in this experiment. But somewhere here, I can clearly say they have similar structure, although I have never seen the structure, just from the sequence comparison. And at some point, this signal will disappear. How can I check where this somewhere is? I need experimental information, yes. But how will you do it, and what type of experimental information will you need? I need sequences, that's also true. Yes?
Maybe do some tests for some sequences, like get the 3D structure.
So I give you some 3D structures. And then?
Then we can see with what percentage really is the same.
Okay, so let me say I give you 10 different 3D structures. Can we do it now? No, we cannot. We need some similar ones. Okay, I'll give you five pairs of similar ones and 5 others that are dissimilar. Is that enough? Are you happy? You're not happy. Why? Because it's a Tuesday morning. or what makes you unhappy? What do you want?
Because we need much more. We have millions of proteins.
Yeah, I have millions of protein sequences. Now let's get back to reality. Initially I said I'm your dream experimentalist. Now I cease to be the extreme person or dream person. I'm now more realistic. I can give you the database. How many can I give you? Anybody remembers the number roughly? How many proteins of experimentally determined structures do we have? Does anybody have a ballpark idea? 55,000, 100,000 is again, what I really expect you to know is a ballpark number. 55 is a completely correct answer. Another correct answer would be 110. So you just give away a factor 2 and you're there. So it's about 110,000. But 55, again, in a case like that, is totally the right answer. If you said 10,000, it's still, we are in the ballpark. If you say a million, then that's the wrong direction of the ballpark. Okay, so I give you something like 100,000 structures. Now what do you do? How do you figure out which ones are similar? Again, you need essentially two bins, or two pairs, or two sets. You need a set of proteins that are not related in structure, and you need a set of proteins that are related in structure. I give you the raw structures. Now what?
We can do comparison and find a match in the database.
What do you mean by comparison?
Sequence comparison.
So you want to sequence compare to find out how good sequence comparison is? Sort of you bite your own tail in some sense. No, you cannot do that. I mean, well, let me slow down. This is what people sometimes do. In this particular case here, because sometimes we cannot do better than that, and when I talked about the scoring matrices, we had an example where that exactly was like that. In this particular example, actually I have much more information. I do not have to get back to this ugly method. I can do something clean. What is it? I talked about methods. So the answer that I want should be part of your experience in this over the course of this lecture. Or put it differently, would this be the exam day? I would hope that you would know the answer to a question like that. I give you 100,000 structures. What do you do with them? I want two sets, one structures that are similar, one structures that are dissimilar. How can you do that? Okay. Nobody? Yeah.
It's sort of like a decision tree or some machine learning where you can say this category is likely to be matched by your...
How do you compare structures? Yes, you can use, so for those of you who didn't hear what he said, you can use a decision tree, and you can, by this decision tree, see what is similar and what is not. But into this decision tree, you will ultimately begin at some point of the root of the tree with a pair. How do you figure out whether a pair of structures is similar?
With a kind of like understanding of the similarity, like edit distance or...
Edit distance, yes, but edit distance is a little bit more sequence comparison.
If you compare like small parts of the strings and try to expand on that.
Guys, this is not about strings. So what he said is if you compare strings, again, what we want to establish is how good string alignment is. And our criterion of truth, standard of truth that we want to match is 3D.
You can watch, you have like example of similar protein structures.
Yes.
We can see how...
How do you have them? I give you 100,000 structures. How do you get similar ones?
15,000 maybe, half percent.
I give you 50,000, whatever you want. But how do you find the similar ones?
You got the similar ones and you can...
But how do you find the similar ones?
You give me a set of similar protein structures.
Oh, no, I don't do that. I'm an experimentalist. I don't know any computer. So you have to figure that is what you have to figure out. I give you the set. I give you the database.
And you simply can classify it.
How? How can you classify them?
Like looking for geometric similarities or amino acids.
Okay, this is the major thing. So I do not expect you to remember exactly the type of methods I sampled when I presented 3D comparisons. But that you can 3D compare, and that is not a string comparison, but somehow a geometric comparison of either objects in 3D where you have RMSD, or you translate the object to 2D maps, and then you compare 2D maps, matrices with one another. That at least should be ready for you when you write your examen. Again, not the details how you do that, but at least the fact that you can do that. Okay, now so we talked about the 3D classification methods that are geometric. So we talked about Voronoi trees, or Voronoi volumes, we talked about 2D comparisons, and we talked about 3D object comparisons through RMSD values. But none of those are string comparisons. That's important to remember. Okay, once I have them, I can simply classify into two groups, where essentially one group so I take the entire protein database, compare all against all. I take the set that has a similar 3D structure. This is some RMSD threshold. I take things that differ, and I have something in between where I'm not so sure. So I leave that out of my comparison just to make sure I go to two extreme data sets and ignore something in between. Now, there's one problem with this. And that problem here is when you start with all the known structures and do an all against all, if this set that you start with had a bias, by doing a pairwise comparison on all against all, you actually square the bias. That's the downside to it. The data set does have a bias. All biology data sets have biases. There are many reasons for that. One reason for the bias in the PDB is, for instance, you determine a structure of one protein. I told you that in the olden days it cost more than $1 million. So now to do another one that is almost the same, or is identical bound, for instance, with a binding, with a molecule bound, this is very important to know for drug design. I do not only need how it looks in 3D, I also need to know how it binds its target. So here we're already down to the level that we're looking at here. We have an identical sequence, but somehow identical structure, just with a ligand or not ligand. For our situation of checking out how good string alignments are, this is irrelevant. Another reason would be, I have one protein in mouse and one protein in human, and they're very, very similar. Again, it's easier to do another structure that is very similar. So I get sort of the credit, and I do a little bit less. So there are various reasons why you would put bias into the database. Some of those are important, such as finding out whether the human protein really is like the mouse protein, or how it binds, and things like that. So we have to find a way to remove the bias. We could do that by finding some sequence-unique subset. And this, again, we would identify by sequence alignment. So we did an all against all string alignment, and we just cluster. How exactly we do that clustering is a different story. It's relatively straightforward as long as we just put the value somewhere, we always improve. And then we take out this sequence-unique subset and compare it against all. And then we're going to have red proteins that have different structures and blue protein pairs that have similar structures. What this one shows is the number of residues aligned, percentage sequence identity, and this is something that Reinhard Schneider and Chrysander introduced, the H-val. This is a curve that somehow optimizes the separation between the red and the blue. Somehow means that most of most of the things that are above here appear to be blue. In order to get now to the safe zone, so what you don't see in these curves, or what is the problem? How to ask that question? So say your method or your plot program had produced these two plots. What is missing here? Yeah?
I have a question.
Yes?
Can you please repeat what is the blue and what is the red?
The blue are pairs of proteins for which we know they have similar 3D structures. So that's what we exactly do by geometric superposition. We have, for all of these, we have 3D structures, we superpose them, and we ask whether by this 3D superposition they're similar or not. If they're similar, they're blue. If they're completely different, they're red. And now I do a sequence alignment, string alignment. So I know the two sets, I know blue and red. But my question is, when I do the sequence alignment, pretending not to know these colors, could I retrieve those colors? Okay? And I'm a little bit more complicated than the zoning I showed you before. In the zoning before, I had only this value. The fact that I show two axes here already indicates to you that with one distinction we cannot really do it. But with these two we do a fairly good distinction. Now, fairly good in the sense that up here there's no red. Okay? My question to you is, can you tell me anything about the density of the red close to above the gray? Are there many pairs that are red above the gray? Can I have a show of hand? Are many pairs, you see a little bit, not a little bit, you see the red here. My question to you, are these many pairs? It seems like yes.
Because the density might...
It seems like yes. Who is with you? Who is with him? It seems like yes. So we have a local cluster here. This is funny. Yeah, is that a question, or is that part of the local cluster on the right-hand side? This is almost so. There's one person on that side who says yes. Everybody else who says yes is on that side. Who is no? That's completely mixed, and is the minority. So the important thing that the important message here is this slide completely suggests that you can make the decision. Be careful. And the problem really is that when you plot these dots, a dot occupies a certain space. And you absolutely cannot see the density. Let me demonstrate to you how you cannot see the density by simply looking at the actual counts. Right? And in the actual counts, so what I'm plotting is actually the distance from this gray curve. So the gray is here, the blue. That's the distance +5, +10, and so forth. And that's -5, -10, and so forth. So 0 was the gray curve. Up here, I write percentage sequence identity. That's just the corresponding percentage sequence identity. Let me go back for this curve saturates. And that's the value that I chose up there. So this is just an approximation. This is the actual value plotted. So the first thing that you see is if we said that the twilight zone somewhere began here, and somehow this line is sort of the point of distinction, then you see that from here to here, blue are pairs of proteins that have a similar structure. There's a logarithmic plot, the numbers here. What you see is that upon intruding into this twilight zone, the blue explodes by more than a factor of 10. So, if I were able to move from this value to that value, I would find many, many pairs that I would otherwise not find. At the same time, what you see is that at the value just a little bit above the 0, actually red pairs of proteins that do not have the same 3D structure are tiny. So what looked on the 0, actually what looked at as many are many. There's something wrong here. I have to revise my curves. So the conclusion from this should have been the opposite of what it actually is. But the major part is another aspect. The major part is that when you go for a value of 8, essentially red is 0, then you move something that is roughly 1, 2, 3, 4 percentage points down the road, and you go from 0 to 50-50. So we are at a point that is roughly corresponding to something like 35% sequence identity. So from 100 to 35, you don't make many mistakes. That's what I meant by the safe zone. Almost no mistakes, very, very, very few. And then suddenly you move another few percentage points, and half of what you find will be already wrong. And that is a cumulative number here. It's not what you find at that point, but it's what you found until that point. So just down here, already half of everything you found from 100 is wrong. And you just move 1, 2, 3, 4 percentage points further, and 90% are wrong. So that is the definition of the twilight zone here. It's the area or the zone in which the false positives explode. In physics, you would call that a phase transition. You have an extremely rapid transition from safe to mistake, or mostly mistake. And that, in fact, is the challenge of the field. The challenge is to push a little bit further because the blue increases so much. And that means we have to come up with methods that improve the string comparison. Okay? How can we do that? One way to improve the string comparison is by going multiple alignments. So I do not only want to know whether two proteins are similar, I want to know whether this protein is related to many others. So there's this protein in mouse, human, rat, zebrafish. Are they all similar or not? If I did the same thing as before, the dynamic programming, so again, I do the complete matrix, and I essentially, for 3, I could do it in 3D. The same matrix. And you can pretty much work out a solution. This is no longer the real optimal solution. But it is, essentially you can work it out for 3. You can even, in principle, rumor has it, you can work it out up to 6, provided you have 60 terabyte of memory, main memory. But protein families are much larger than that. So this is not enough. Because this is not enough, we need a hack. One hack is we do some pairwise alignment, and in the intersection of the pairs we do an n-wise. So somehow we would go, in this example of 6, we would go from 12 where we have pairwise, or depending how we merge the pairs, to a little bit more. A more extreme hack is the one that Rost introduced, and that is essentially doing it all pairwise. So you begin with doing A against B and C against D. That would be one way of doing it. And then you essentially pair them up. Pair them up. Now there are different ways of doing that. One is you essentially write a consensus sequence. Consensus sequence is you take the letter that appears in both. So beginning with this string here, you essentially take whatever is common to the first 2 to do the same thing here, and then in the third step you would essentially align the pair of pairs, which are now no longer real sequences. They are sort of the merger between these two, the merger between these two, and then you align the 2 mergers. Okay? This is the first hack. Is the idea clear here? Okay, let's begin again. So far we figured out how to compare pairs. So I can compare this one with that one. What I'm going to write here is somehow let's call it the dominant or consensus sequence, the thing that describes both of those best. It's not an actual protein sequence. It is one wherever they are conserved, I take whatever is conserved, and when they're not conserved, I have a variety of decisions. I can take the most informative one, I can take the one from the first. There are different ways of doing this. Okay? But this gives me a new sequence that is neither this nor that, but describes both of them in a somehow good way. Okay? I do the same thing for the second pair. And now, actually, I compare these 2 pairs of pairs, or these 2 consensus sequences. Because again, I do 1, 2, 3 pairwise alignments. And pairwise alignments we know how to do. And essentially what I get is an alignment of 4, or hack for an alignment of 4. Is the idea clear now?
Sorry, but why is the second step required?
So the second step, again, this is sequence 1, 2, 3, 4. Or
These pair of sequences belong to different species.
yes, I'm sorry. I should have gobbled it up again. It's amazing how I can gobble these things up. In particular, the easy ones. So protein A, protein B, protein C, protein D. They could be from 4 different species. And I believe, just imagine they would be from 4 different species to begin with. I want to align all 4 of them. I do not know how to do multiple alignments for 4. So I break it down into pairwise problems, because pair I can do. One way of doing the pairwise breakup is I do A against all of them, and then I find A first against the most similar one. So that would be B. That's step 1, shown here. The next most similar pair in my list Well, A against B is already given, B is already aligned, so C and D would be sort of the next most similar. Shown here. So far, so clear?
Why can't it be B and C? Because that is...
It could absolutely be and C. So there are different ways of playing that, and I'll get to that in a minute. But this is one way of doing it. I'm not saying this is the only way of doing it. This is one way of doing it. Okay, yeah?
Why are we only comparing 3 sequences or 4?
Now again, so in the first step here, I do A and B. In the second step, I do C and D. So now I have, in the second step, I have used all 4 sequences. I have not put them all together. I have essentially done 2 independent pairwise alignments. Right? AB, CD.
What does the third line mean in step 1?
The third line now is the consensus sequence. So I'm going to look in this alignment here. What is the residue? Is there a run residue that both of them share? It's the G. In some cases, I will not have a shared residue. So here I have to make a decision. Do I now write an E or D? Or do I write E or D? Or do I put a probability? And essentially, I could do all of those. The simplest one is I pick whatever happens most, or whatever is the most informative, or whatever is the first. Let's play it here, whatever is the first. So I write down the E. What I get here is a pseudo sequence. In this particular case, actually happens to be the same as the first sequence in my example.
We compare 2 random proteins.
Yes, you could call them 2 random proteins. These are 2 proteins that you find are very similar. In that sense, they are not random. They're 90% sequence identity, most likely by what I showed you before. They have a similar structure. But now what I want to do is not only pairwise pairs of similar structured proteins, but an entire family. Families I cannot build up because I cannot do an alignment of an entire family like that easily. And here's one hack to build the family up by pairs. Okay? And again, this is pair 1. Protein A and B are aligned and create this so-called consensus sequence. In step 2, and again, this is one way. There are other ways of doing that. In step 2, I could, for instance, align C and D. And then I really have essentially built up, have somehow involved all my 4 sequences. So that's the alignment of C and D. And again, I write down this consensus here. Yes?
Do you understand right that in the protein that we take the last one, it's one of those that belong to C or D, that is not something completely else?
So in this particular case here, it's true. I now see some limitation in the way I construct the example. So in the way I do it here, for simplicity, this will always be identical to that. So I have to sort of come up with a new rule in order to change that. The new rule could be I take whatever is the highest probability or the most informative of these 2. And that could be decided by the BLOSUM exchange matrix or something like that. And then this, so sometimes simplifying examples kills the message. And I'm afraid that I did another killer here. Because indeed, yes, that is identical to that one, the way I constructed. If I define take always the one from the first, if they differ, it's the identical sequence. So it's a little bit of a dumb example. But you could imagine that I replace this by the most informative one, and that my example is chosen such that just it happens so that the E is more informative, and here the L is more informative. Or here the I would be more informative. Okay? Then I would actually have 2. Okay, I still need one additional difference in order to make the example good. Are you still with me? So in this particular case, this is the first, that is the first, but in a more general case, if I used a different, so I could put more differences here, and in one case it chooses the highest probability is this, another one it would be the lower one, and immediately see that would be a mixture of the two sequences. Is everybody clear about that? No? Does anybody say no? I'd like to stop, not to go on before, yeah?
If you have the, I mean, if you pick a rule that we are going to take only the first one,
yeah. So,
next one, can we do the second one?
yeah, no, that's exactly, my simple rule didn't work. So let's assume that the example would be G, G, Q, L, A, K, E, E. So I'm just writing what is there. I'm just changing the second one. And the second one is G, A, Q, I, A, K, D, E, A, L. And I now pick the most informative one. And here, say it would be the D. And there it would be the L. And there it would be the A. Then, and all the other ones, I pick exactly what is identical. And you immediately see that I now create a consensus sequence that is neither this nor that. Okay? Okay, so then I have now two objects, or two sequences here, that are neither the first nor the second. Is there a question left?
Question about the numbers in the table.
The numbers in the table essentially means, so I have to somehow start somewhere. Indeed, what I show here is one solution. You could do it differently. But one way of starting is, I first do A as a pairwise alignment against all other three. And I pick first the pair that is the highest identity in sequence. The most related pair. In this example that I show, that is A and B. Indeed, I could also start with B and C. This has an equal number here. That means I have to make a choice, and in effect C and D too. But the choice A and B is as good as any other, and that's the choice I take here. The next most related then would actually be C and D. I made a mistake on saying that. This is what the numbers mean. And again, I could do it differently. But this is the most reasonable way of doing it. So essentially, your idea of building up a tree would work exactly that way. There are different trees we learn immediately from this choice here. So you could you root your tree in a different way, but leave the tree in a different way. There are different trees, but this is one of the trees. Everybody clear about this step so far? Is there anybody who is not clear about it? Good. So then, again, my third step simply is I have two sequences. Again, they are no longer real sequences. None of the two in a real example would be identical to any one I started with. And now I do the pairwise alignment of these. Same thing, strings, same letters, all the same rules. Okay? And then ultimately what I get is a pairwise alignment. Another way of doing it, and again, I'm not going to show you all the ways, but just to show you, there is immediately another way of doing it. So first you could actually build this consensus sequence of A and B, and then say, now I go for the next one, C. So I essentially build up my alignment. So in the first alternative, in the first one I showed you, I do pairs of pairs. And here's sort of build up. And if you want to build up a family alignment, somehow the build up is maybe exactly the kind of thing you have in your mind. Ultimately what these pairs do is they allow you to go further into the twilight zone without making mistakes. Now, the next issue is the one of using profiles. The concept is the following. We again build up pairwise alignments in different ways. Building up pairwise alignments, we may end up with something like that. Looking at something like that, And again, I see that my example is maybe not very well chosen, but the point is that it's very poorly chosen, I have to admit. So let's take the next example. In this example where I'm without letters, you immediately see that there are regions that from the comparison of all the sequences, you see that this you somehow should align. This you somehow should align. This one, there's no, the sequence is shorter. And here you can leave something open. And that contains information. And let's begin with the first idea of it, and those slides are from Teresa Word. The first idea is you simply, in this particular case, example is for a transcription factor binding sites for DNAs, in this particular case, therefore only 4 letters. The height of the letter is proportional to the number of these counts of these letters you find at that particular position. The transcription factor binding site is at 0. This is left of it and right of it. And you see, for instance, at position 6 and -7, you have a lot of C, -6, you have a lot of T, and so forth and so on. Now, you can use this motif in a regular expression. This sort of thing you have all seen with grep or egrep or something like that. And these are sequence motifs. So simply, at some position you could have an L or an I. At some position you have a charged residue, and then there are a certain number of axes you can have in this particular example here. And you can build up these motifs. Then some of them are not allowed. And essentially now you can ask, when you look at the next one, how do you best fit that motif? Okay? Very simple. You can do that in sort of in a regular expression sense. And you can do it in a probabilistic sense. In a probabilistic sense, you would begin with something that is sort of a generic scoring matrix, and you would evolve into something that is a specific scoring matrix. So the generic scoring matrix says I can exchange the tryptophan. If I match the tryptophan, that's very good, remember? Or that some residues are hydrophobic. I want to match a hydrophobic and a hydrophobic residue. That's sort of the generic statement. But for that particular protein, I can have a specific statement that comes from the counts I see at that position. So on the example of the nucleotides, and I didn't remember that there's an animation in here. So this is my alignment here now for a sequence of 4 nucleotides that are 6 letters long. And then you simply count on the first position what's the absolute frequency. You turn that into a relative frequency. Sum is of course 1. The relative frequency gets into a log-odd table, and that log-odd table you then use for the alignment. Meaning that, oh, sorry for the animation, I have to get the animation out. So meaning that now, next time, position 1, 2, 3, 4, 5, you can essentially exactly estimate what's the probability of putting an ATGC there. Right? Your next step is you have to find a way to sort of align according to this formalism. How could you do that? Exactly. You do essentially. You remember I showed you the scoring matrix that we use in order to find the word matches in BLAST. We replace that by this one. That's all we do. Just the same matrix. It's only position specific. Now the additional problem somehow is you have one of these matrices per position. But that is exactly what a profile-based alignment means. So this is such an alignment of a large family. In this particular large family, you see that 1, 2, 3, 4 columns are largely conserved through the entire family. And once you see something like that, you know whatever you do, those columns you have to maintain. They are the most important ones. And at other positions you have certain things you can put in there and certain things you cannot. So on your 0 step you would start with the generic matrix, and then you would sort of replace it step by step with this specific scoring matrix. And that in fact is essentially the concept of a method called PsyBLAST. You begin with a fast hashing. It's exactly what we had before. So you find identical words. in the first round. So you take your sequence, look against the database, do the fast hashing, just what we had before. The tiny bit of a difference now is you in between, you extend by dynamic programming. Again, dynamic programming, I said, is slow. Why can you afford to extend in between by dynamic programming without getting too slow? You find word matches up here. In between the word matches, you do dynamic programming. Why can you do it now and not for the entire sequence? What has changed? What's the difference between doing a dynamic programming on the two sequences up there and doing a dynamic programming on this part, for instance?
There are no matches.
Yes, but this is correct. But you want to find out whether it's true. So you have to run a dynamic programming in between.
To be possibility of different structure. To be possibility of different structure, if we merge this big again.
I believe I'm unclear in my question. I'm not repeating your answer because I believe I learned from your answer that my question is not clear. So again, we begin breaking down my match into fast word matches. I cannot do dynamic programming on the top because it's too expensive in terms of CPU. After the breakdown, I claim now I can do the dynamic programming. Why? What has changed? Yes?
The sequences are much shorter.
Much shorter. Remember what the order of the complexity or the speed of the dynamic programming is? It's the square of the length. So the moment I break this into 3 parts, I have 1/3 times 1/3. So I have broken it down by almost a factor of 10. So the point from the breakdown is that so essentially the speed is square of the length of the comparison. Even if I halve the sequence, but if I have only one hit in sort of roughly half of the protein, even then I quarter already the time. And in a typical run, so what I do is I find my initial hits, I extend them a little bit, and then I run the dynamic programming. So I always break it down to much more. There's more than one order of magnitude faster in a typical case. Okay? So the major thing here, since dynamic programming is on the order of the square of the length, break down, finding matches that break down the sequence into fragments helps you tremendously. Okay? This is the square effect. Then you do again the comparison against the background and you get an e-value, expectation value. You collect all the pairs. So again, you do one sequence against the entire database. And you do that pairwise. And say you find 100 hits. 100 hits are such that 100 cases are clearly above, way far away from the background. Okay? Now what do you do in the next step? I have 100 pair hits. And what I want to get is a position-specific comparison. I want something that does not only look at pairwise comparison, but looks at a family. So I have 100 pairs. I told you the hack to get from pairs to a family alignment, right? That's what you apply next. And then you just align them up, and what you get is this profile. And this profile is giving you a probability for at every single position matching, in this particular case, a Y, for instance. Okay? Now what you do is you take the profile and run it against every single sequence in the database. Because the profile is more precise, or it allows you to look deeper into the twilight zone. Now you may find things in the first 100 you may not have found. You're more sensitive. Okay? So you run again. And what do you do next? So the method is called PsyBlast. PS stands for position specific. What could the I stand for?
Iterate.
Yeah. So you do it again and again and again and hope that it converges. So the simple criterion for convergence is that you don't find anything you haven't found before. And that indeed is one of the most powerful tools in molecular biology. So the word blasting has become a verb. There is no drug that has been developed without using blast. And today when people say blast, they always mean this psi-blast. Every experimental study, almost every experimental study in biology begins with running a psi-blast, with finding whatever new protein you look at, is there anything in the database that is similar to it? And what can I learn from that similarity? It always begins here. Okay, so that's the iterative step. We could do this in other ways. So psi-blast, in fact, was not the first method that established this idea, and it most likely is also not the best that does it, but it's certainly the one that is by far most popular. And one of the reasons is because it does a very good job and does this very good job very quickly. So here is cluster W. That's a slightly different type of method, and there is an entire generation of cluster W going from 88 to 2011. But I'm going to gloss over that, and just the major idea behind cluster W is simply exactly what I showed you before. You do an all against all, you fill in this matrix in this protein 1 A, B, C, D. You do that by a dynamic programming, and then you build up a phylogenetic tree, and you use that phylogenetic tree to do an alignment of pairs against pairs. Not with consensus sequences, but essentially with profiles with probabilities. That's the way it is done. It is highly cited, has hugely over 100,000 citations at this point. The problem, so essentially Cluster, in doing this here, builds up a family from pair comparisons through dynamic programming. So it's somehow more accurate than psi-blast because it always uses the optimal method. It's the problem, of course. What do you believe is the problem of this? Slow. So today, where we have families that can go to 100,000, so this is really getting very, very, very difficult for cluster. And the immense achievement is that there are now computers that can, in fact, run cluster alignments even up to 50,000 or something like that. But some families are more than 100,000, so cluster cannot handle that. Another idea is presented here as Maxom, is one of the earlier methods. And the big issue here is simply focus. So the focus of Maxom is that it argues, well, I begin my alignment with a particular sequence. I want to learn something about that sequence. That sequence I don't want anybody to touch there. I don't allow any insertion in that sequence. That sequence is real, that sequence is dear to me. In the family alignment approach that we had so far, we transferred, remember we did A to B and C to D, and we did consensus sequences. And none of them, at the end, you did not have any preference for anyone in this group. It could have been, so the lettering ABC, it could have been completely the other way around. Now the big issue really for Maxom is to say A is the major. So when I build up my pairs, I always do AB, AC, AD. It's always A against the universe. Not universe, against the family. To show you an example, the protein-centric view here of psi-blast and Maxom would be like this. And the family-centric view here of cluster would be like this. So the sequence A here does not have any dot in it. That's the major issue. Okay? Yeah?
In the Maxom method, how do we arrive at which sequence is A?
So the question is, in the protein-centric, in the Maxom view, how do I arrive at protein A? So how do I declare this is A and this is B? And ultimately the answer is the user does. So A is the protein you care for, or the user of that method cares for. So when applying that, it could be, I want to know whether that protein is related to something else. I want to know whether that protein binds DNA. I want to know whether that protein forms a particular 3D structure. So it's always that protein is centric that is the beginning of my study, that I care for. While here the idea is that somehow I want to study a family, but not any particular protein in that family. Okay? It's sort of a tiny difference, but a very, very important one. Now, the other type of methods are HMMs. We get into that in a minute. So again, what sequence profile methods do is they go into the twilight zone, much more into the twilight zone. And profile-based alignments, what you have to do again, you begin by sequence, the result is a profile, then you do profile sequence alignments, and the result of that is an updated profile, then you iterate. So the other way is the hidden Markov model. Does anybody in the room here, has anybody ever encountered hidden Markov models? So what's the major assumption about a hidden Markov model? It's not directly spelled in here, but what's the major point?
We use probabilistic approach.
It's a probabilistic approach. That is a very important point, yes. But there's something that comes ultimately with the name Markov, yeah?
Memoryless.
He says that it doesn't have memory. Not quite. It memorizes one step. It just memorizes. So every decision is based on the last step. You may make the argument the last step is not, I don't need memory because that is my other foot. Fair enough. But so essentially the Markov process is one where in the time step t is influenced only by t minus 1. Or whatever happens at position I is only influenced by position I minus 1. Okay? And so essentially what the probabilistic part of it is, you start with a state and then you have a transition to itself, or you have a transition to the next state. And then in this particular case, you have an end state of the hidden Markov model. The transition states somehow here are the observed symbols. The original states are the hidden states. Let me give you an example. And again, those slides here from Marco Punta. Marco likes to play football, and he did that at some season in 2014 or 13-14. For some particular football club, he looked at what is so this is the club he looked at, what's the score playing against that club here, how many people were there, and so this is all the Bundesliga. And so the wins are in blue, draws are in black, and losses are in red. Now, you can essentially put this in lose, win, draw. That's what I showed you. And you have essentially a probabilistic model. So going through that table, you can simply look up what's the probability that at one point you have a win, but at the next one you will have another win. And the model is, essentially you have those three states, you have a simple probability. Now, what else do you believe you could use as information to predict? So far, it's a simple probabilistic model. There's a certain probability that after a win you have a loss. Is there any additional information you could possibly use in this table?
Frequency of win or loss of a particular team?
The frequency, that in fact, so let's get back, that's an important point. So she says you can use the frequency of winning, but that is exactly what your null model will do. The null model, and again, thank you for pointing this out to me, I'm too fast. So the null model, the simplest thing, when you compile the probability, so I win at one game, what's the probability of next time losing? So the null model is simply, I take the table and I look up the probability, and I look them up from the frequency. Okay? That's the simplest I can do. Right? Is there anything that you can do that is somehow equally simple, but comes as the next complicated thing? No, I cannot see whether the next slide has the answer. Yes, it does.
Probability of winning at home?
You saw the next slide, or was that your idea? Okay, so you can simply classify them according to home and away. Some of you may see sport events, and indeed it is the difference whether the teams play at home, in particular stadiums. So now you would have home and away, and that somehow in this particular case here is the hidden variable, or it's the hidden information. It is the part of additional information that you hope will increase your prediction. Now you have a coupled probability model. And you can immediately see how that can help you. In terms of sequences, again, you have a transition probability. In terms of sequence alignments, you write everything here, the states and the symbols, as a discrete probability model. What about this one here? A traffic light. Is that an example for a hidden Markov, probabilistic hidden Markov model? No, it's not. And it's not because it's completely deterministic. You absolutely know what will happen in the next state. Again, at least once you have observed this traffic light a couple of times, you can clearly sort out the numbers, completely deterministic, absolutely not probabilistic. What about this one here? So the weather prediction, it's sunny, cloudy and rainy. Yeah?
Yes.
Yes, that is highly probabilistic. In fact, it is even complicated to do that on a Markov model, on a first order Markov model, but this would be in principle the way of doing that. Now for an alignment method, if I wanted to use that for an alignment method, I would essentially every single time in the model have a match state, a deletion state, and an insertion state. So I align, essentially now I build a model and I ask the probability of having a new sequence that aligns to that model. The model, does that sound to you like pairwise alignments? Okay, you have two possible answers. Could be yes or no. Abstain is not an answer. Yes or no? Who's going to defend yes? Does that look like a pairwise alignment? Yes? It does. Do you want to defend it or want somebody else to defend it? Somebody else wants to defend it. Who wants to defend yes? Yeah? You want to defend it or you want to just say yes? No, I want somebody to defend yes.
Insertion and deletion of amino acids based on the evaluation. Yes, it will.
Yes, we have insertions and deletions based on what. But if you think about it as a pairwise alignment, where do you get the probabilities from? So whoever says yes to that has to give me some idea where we get the probabilities of transitions from. Yes. Yes. Yeah. Okay. You're totally right. But so now I want to map the problem pairwise alignments upon such a model. I define my model as in you have match states, so the number of match states here are equal to the protein length, and I want to map the problem pairwise alignment upon this. And can I do that? The answer is yes. Yes, you can. But if you can, my question to you now is, what corresponds to the transition probabilities? Yes?
Like the sum of the weights going from the match states?
The sum of the weights, well actually,
probabilities, like
the probabilities, where do you get them from? If you want a pairwise alignment.
Like if you give the model a lot of strings.
Yes. Then you can do it, but I'm asking you to give me the string. So where can you pick up, so what's the sort of null model? Pairwise alignment is in this image a null model. So null model means the first iteration, if you align pairs, the first initialization, if you want, of this model are the transition probabilities, and where would you get them from? Anybody?
Experimental data?
No experimental data. Somewhere in the database, something that is on some of the slides that I gave you. Yes, exactly. The generic, whoever said that, the generic scoring matrix, any one. The one I keep showing you is the BLOSUM 62. Whatever matrix you have, this is the null assumption. If you don't know anything more, I told you this is better than identity matrix. You could put an identity matrix into this one too. So that gives a probability of transition to the same amino acid, and all the others probability is very low or something like that. But there's the scoring matrix, you can translate the scoring matrix into a transition probability. Yeah?
But only for the matching, right? Not for deletion.
That is a little bit of a problem. You're totally right. So you can only do that for the scoring matrix does not contain open and gap penalties. And the formalism of the dynamic programming, I always told you, you need to provide a parameter for that. And you could have a generic parameter. So you could initialize all the deletion and insertion states with a sort of certain value that is again taken from typical dynamic programming. And the important thing is that the value of the relative penalty to the transition probability. Because you take the scoring matrix, you translate it to probability, then you also have to sort of translate the gap penalty. But that is trivial. And again, it's the 0th answer, right? Now, once you have a pair of related things, or a bunch of related things, you can immediately see that every single time you add to that model, you can refine the transition probabilities. You essentially can learn your data. Is that clear? So you begin initialization with a standard scoring matrix, and once you have a family, every sequence you encounter, family is defined as things that you believe are related. So now what you want to build is a probabilistic model for that family of related things. So every time you add a sequence to that family, you learn that sequence, you improve, you update your transition values. This is like in PsyBlast building up this iterative profile. Okay? It's essentially the same procedure, except for now we have a sort of entirely probabilistic mechanism. What I did not say before, I'd say, no, let me get back into that later. So this essentially, you begin with something where you have simple mesh states. The 0th level is essentially that. You start with a single sequence, you build a model from the typical background levels. That is simply, you increase reduce the entropy. The entropy is maximal if you have a variation, and is minimal if it is completely, is it most informative. You re-estimate the alignment, you build a model, you iterate through this like through the PsyBlast. In principle, the idea is clear. The details of the methods are highly non-trivial and would occupy much more than, well, now I give you 1 minute. Understanding how the entire HMM algorithm works is certainly more the kind of thing for a couple of lectures. What is important here really for me that you understand, this is a probabilistic model. The idea behind it in principle is the same as the PsyBlast, and you can sort out the details if you had time for that. But, is there any questions so far? Yeah?
If you have trained a model and you shoot another protein at it, like how do you do classification of like what's possibly in the group and what is not?
So once you have, I removed the last slide and that's exactly what I need now. So once you have your model being defined as you have all the transition probabilities, the insertion, deletion states, now you come with a new sequence. You simply compute the entire probability for that sequence and find the most probable model, most probable fit to that model. As simple as that.
But
there's one solution.
But how do you get to one value, like one probability value?
You search the space of all possible paths through that model. That's a Baum-Welch algorithm. It's not that trivial, but fairly straightforward. And in fact, this is a little bit slower than the PsyBlast once you sort of align into the model. So building up the PsyBlast is slow, building up the model here is slow, but the last step is simpler than it sounds. Because for any single sequence, you have actually a fairly limited number of steps through that model. And that makes it fairly straightforward. Now, let's get into genetic algorithm. And the genetic algorithm, important background here, is in everything I talk to you about essentially fulfills the Markov condition in some sense. The Markov condition being that when you look at two different positions here, they are completely independent. What happens at position I is independent from what happens at position J. This is the only that was the condition for the dynamic programming to find the optimal path. That was the condition for the BLAST, and that is the condition for the hidden Markov. So you have a dependency only on the previous step. So columns that are far apart are independent. Now in proteins we know they're not independent. Why not? There was somebody said something here. Evolution, that is true. But there's something, even if there wouldn't be an evolution, there's a correlation between sites. Between this site and that, for instance. Between sites that are not close in sequence. What constrains them, or could constrain them? Not all sites that are not close in sequence are constrained, but some are. Can you think about something that constrains two positions in a protein that are not close in sequence? So in the example, this and that, for instance, or even further away, yeah?
It's not possible to construct a protein with like a special distinct sequence.
This is true, so let me turn that around. In order to follow up a protein, you need to bind. And binding is done by contacts. And the contacts are not local. It's a little bit longer to argue why. You could actually not imagine a protein to be built up only by local contacts. So I and I plus 1 or something like that. In order to form this into a compact object, you actually need to introduce long range, so more than I plus 1 or more than I plus 3. So contacts are what you maintain, right? So these two parts here bind, and if you want the same structure, then the next one has to have this binding constraint. And that means the initial condition, I and J are independent, is completely violated by protein structures. Yes, also in evolution, because evolution fulfills the constraints of protein structures, but the physical reality that violates it are the structures. I need to maintain a similar 3D structure. The 3D structure needs non-local interactions, and I have a constraint. And that constraint is dependence between I and J. Not between all I and all J, but between some I's and some J's. Right? Clear? So, whatever I produced so far, in fact, has a very limited reality in biology, because it completely ignores the absolute essential correlation of positions that come close, contacts. Nevertheless, essentially all sequence comparisons are done with this bad violation. Essentially, this completely unbiological assumption fits the model fairly well. All alignments, most of the alignment methods use it, and I don't know, 99% of all the sequence alignments that are done today use this violation. So it works still pretty well. Here's a method that could in fact break that down, and that method does work better and allows you many things, but has another disadvantage. It's a genetic algorithm. It doesn't make the independence assumption. It essentially operates on segments. So one example that you find in Wikipedia is this antenna that in fact has been optimized by a genetic algorithm to fit a particular radiation pattern, to be particular. It's called an evolved antenna. You may not immediately see how that differs from what you would do when you're bored, but this is an evolved reality. Now, in the protein, how would you do that? Well, you would start. You would initialize the population, you would compute a fitness, not in the protein, so in a general sense. Given a certain level of fitness, you have a stop criterion, or you begin to sort of roulette parents to cross over to children, to mutate the children, and then you compute again once you have gone through one of these. So the logic is not quite right here. So you compute the fitness once you have done one or all three of these steps. And then you get back into the stop criterion. You may go to end, or you may repeat. Principle, idea is clear, right? Everybody clear about that? In principle, crossover, simple. Now, In the world of protein sequences, what you would start with is, in principle, I told you, there are different gap penalties. There are different substitution matrices. There's an issue between global and local alignment. And this is exactly what you begin with. If you want to do an alignment, genetic algorithm based on alignments, you begin with a variety of differently created alignments. Okay? The more the merrier. Then you have your genetic algorithm extension and you create a multiple alignment. The beauty of the approach is that you can not only look at a bunch of different alignments and find the best path through these different alignments, but you can actually also use particular information, such as structural information, constraints that you know from somewhere that these two residues have to be close in space. It's totally easy to put that into that formalism. It will just, in the evolutionary algorithm, in the stop criterion, at the end of the day, you will find the optimal solution. You can put any type of criterion in here. In the sequence alignment, you cannot. In the formalism of the HMM, impossible. In the formalism of PSI-BLAST, you cannot put contacts in. You can put additional constraints, but they no longer allow you to optimize it even on the level of pairs, not even, and certainly not on the level of entire families. Any constraint here ultimately will evolve. What's the price you pay? Yes. So this is very, 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 very CPU intensive. This is many orders of magnitude slower than Clustal, and Clustal already is many orders of magnitude slower than PSI-BLAST. So this is a surefire way to use the SuperMOC for one big family. There are many other methods, and some of them use sort of the PSI-BLAST kind of approach of fast searches. Some of them use in speed up on the dynamic programming. But ultimately, the ones I talked about are mostly used today as PSI-BLAST, followed by hidden Markov models. What these methods can do again, they can go into the Twilight zone. Is there anything that is more fancy? So if you look at the evolution, let's call it not the evolution of biology, so the evolution is the wrong word, the development or the advance of alignment methods. So we begin with pairwise, we get to multiple, we get to sequence profile. What could be the next step? I heard the answer. Can whoever said that a little bit louder? I believe it was the answer.
Profile.
Yeah, a simple extension, right? So in this particular case, what you do is rather than taking a sequence against a profile, you compare two profiles. I said that 4 entirely conserved columns are important to describe that family. Now we have 2 families that we describe, and we're not matching one of these sequences into that profile, but we compare 2 profiles. In principle, it's totally trivial. Now, in many cases of computational biology, some of the solutions are very simple and get immediately complex because you cannot decide on alternatives. And here, the problem is you have too many free parameters. The first one is as trivial as how do you compile the vector overlap between 2 profiles? 2 profiles are 2 vectors. The matching of two vectors you can define, you said before, edit distance. You can do a Hamming distance. You can do a Hamming distance on similarity matrices. You can do Hamming distance on a variety of different things. And it immediately gets into a complexity that is not easy to handle. The other issue here is when we look at vectors alone, we lose some information. What I mean is just stare at this, let's call it family alignment. If I compiled just a value between this vector and some other vector to look at the profile alignment, what would I lose? So I'm going to compare, in profile, essentially I compare this vector to another vector in another profile. But what do you see here in this example that you lose by comparing these 2 vectors, or by a typical simple comparison of these 2 vectors? Yeah?
The rest of the profile.
That's true. But I meant something that is specific to that position. You lose, so let me tell everybody else who cannot hear it. So by just looking at two columns, I lose the rest of the profile. Totally right. And this is back to the genetic algorithm that essentially looks at segments. But let's get back to a method in profile comparison that really ignores everything beyond the position I. Yes, I lose the rest, and that is bad, but every alignment method does it, and it's still an improvement over sequence profile, so let's just lose that information. What else do I lose? Yeah? Was there another answer? Do you see something here? The connection to before and after. What do you mean by the connection to before and after?
TNL from R. If you take that vector, you don't know that all of them are connected to TNL.
Yes, that is true. So you don't know how you build up the profile.
Yeah.
That is correct. But it's much simpler. So ultimately, think about the vector comparison. What the vector comparison will ultimately say, there are 3 R's, 3 K's, and 2 W's, somehow. What you ignore in this is that yeah?
Maybe the order?
Yes, you ignore the order. What you completely ignore, even when you compare to vectors in Hamming distance, you maintain the order. But what you ignore somehow is the correlation in the order. So when you see something like that, in particular when you see the other side here, but even when you see something like this, you would suspect that they typically in biology we order them by the first two are most related, so decreasing relation. Then you begin to see that this is sort of a subgroup. This is a subgroup, that is a subgroup, and that is a subgroup. And that, by comparing the vectors, is very difficult to maintain. Because you only, I mean, at least there's no simple vector comparison operator that does this without first clustering. Okay, so I said that for the profile alignment we have an explosion of possibilities and we don't quite know how to solve that explosion. But on top we have a problem that we may not always want. It may be that we are extending our comparison too far and that what we are comparing loses important information. Let's talk about the sheep hack toward profile, and came from Dariusz Przybylski, and this one way to sort of find a way between the one that is CPU intensive and complicated to do profile and profile sequence. Can you think about a hack? Using something that I already introduced before?
Cutting the vectors to segments?
So you could cut it into segments. You mean like the BLAST does it in the hashing? That essentially would be one way of doing it. But the segment still is not a profile. Also, you would do profile on the more local profile. You could do that, but it's still profile. So for every, so you cut it down into a segment, and for that segment you would still do the vector comparison. So you don't win. You still, okay, you reduce the problem by sort of localizing it in some sense, by constraining it in terms of, true, is an improvement. Okay, so you got down. So there's an idea with which you certainly get down. True, but I mean more. Can you map it? Can you map a profile to something that you can actually do, and that is more similar to what you do? So, anybody? A sparse matrix. What do you mean by that? This is a perfect interpretation of the answer that is on the next slide, if you give me the right meaning. Well, that is a profile alignment. In some sense, you do a similar approximation. You throw some away. You essentially put a filter into a delta filter or something, a high wave filter or something. So he says local, you say strong signal or something like that. That would create a sparse matrix, but it's still local. That's not what I mean. Yes.
Count the number of amino acids in the, like in profile sequence.
those which are not matching, count their number in a matrix and then...
Yeah, that is one way of simplifying, that is one way of creating a sparse matrix. But in a large family, you will not have that issue. You essentially will, well, yes, that, okay, fair enough. Is a solution, yeah?
Building sub groups, basically, and doing like the trick with the pairwise comparison done in the sub profiles.
Yeah, so that is not a reduction. That is a great idea, and we still haven't found a good way to do that. So that is a great idea. So in effect, that great idea would be on top. The idea here is cluster your group into different subfamilies and essentially do all subfamilies differently. And so some people have tried this hack. And it does, so we have particular examples in particular competitions where people do this hack and it works. But there's still no good formalism to put it together in a method. It's a great idea, and it's still, that is one that remains to be somehow addressed in a more thorough or systematic way. The field hasn't been able to do that yet. So let me give you the answer that I mean. It's consensus sequence. So what we do is we build up the profile. So those are this is the running sequence. This is the 20 amino acids here, and every single position. So you do the last alignment, you build up a profile at every single position, 1, 2, 3, blah blah blah. You know what's the most likely residue in that position. The most likely residue in that position here would be the L. In the second position would be the S, and so forth and so on. So what you write down here in the last column is exactly the most likely residue. Why is the second one? Okay, I overlooked the P. So you write essentially the most probable position from the profile as a residue. So now you took this profile in its complexity and wrote it as a sequence. It's a hack. Again, in some sense, it's a sparse matrix hack, if you want, and in some sense, it's throwing some away. It's the opposite of extreme of throwing some away. You throw all the 19 away, other than the highest number, and that's the only one you take. So, in some sense, your ideas were aligned in that, and then you have a sequence. Now, how do you do the proper profile alignment, or the pseudo proper profile alignment? That is the double hack. Yes?
And then pairwise alignment.
Yes, that is a double hack. And you're back to a trivial thing. We already know that sequence against profile is better than consensus sequences against consensus, sequence against consensus sequence. So we essentially do consensus against profile. Okay, you're totally right. You actually hack a step further. Hack a step further means you speed up even further. But we can map it to another problem that we already solved, which is sequence against profile. So rather than sequence against profile, we are looking at consensus sequence against profile. But there was a question left here somewhere. Is that cleaned up? Cleared up?
So, I have a question for the numbers. Is it like just the probability of how often in the family, in the first position, is the L, is the A, is R, or what are the numbers?
In the example of the hidden Markov model, that would be exactly, the answer would be yes. In this particular case, in this position-specific scoring matrix, this is a value that reflects something like the probability, but as you see, there are negative values. So this is the one that is used for the scoring matrix for dynamic programming. And for the dynamic programming, if the entire matrix has a slightly negative value, that is an advantage for the speed of the algorithm or the simplicity of the algorithm. So that's the value. You can turn that into a probability for the residue to be there. Yes? You do the consensus. And again, this consensus against profile is somewhere in between the two extremes. In terms of performance, so down here you have the native pairwise blast, here you have the psi blast, that is sequence profile, and essentially what you have here, false hits, true hits, what you have here, so the higher you are, the better you are, that's the consensus against profile. So it gains almost, so the jump from pairwise to sequence profile is almost similar here to go further. You gain a lot, and again, it's only a hack. The most modern way for the profile version is in fact also a hack. It's called HH Blitz. This is essentially the entire system that does it. Ultimately, what HH Blitz does is something like what I showed you before, except for replacing the idea of a consensus sequence by an object that is no longer a sequence and has a variety of features. So you replace the amino acid sequence by a local feature system. And then you do a consensus on the local feature system. The local feature system in this particular case is an alphabet of 200 symbols. But in principle, whether you have 200 or 20 is no difference. So everything else is the same. And HH Blitz worked out how to do that in an extremely fast way, such that these profile comparisons are really available for everyday comparisons. So HH Blitz now can use a large cluster and essentially update the database every 3 months. So that's the amount of CPU time you need. If we have a couple of thousand of processors, they run for 3 months, and you have a new version of the system. Once you come up with a new sequence and compare it against all the database, that is fast. But for that, they need to do all the profile base, or update all the profiles in the database. And that's the tricky part. So with the profile, you can really intrude into the twilight zone and ultimately get into something like the midnight zone. So here's the word midnight zone. The word midnight zone, now what I take all the proteins of known 3D structure and I compare them. And I ask, how similar in sequence are they? And when I do that, I see, and the bar here means there's something that does with happens with the statistics, with the counting. The numbers here to the right should be lower than they actually are if we compare the same thing, but then there are some issues here. But ultimately what you see is the vast majority of all the pairs of protein structures that are similar in 3D have very little sequence identity. Around 10%. So what I show here is the random. So essentially this is 20 amino acids, 100 by 20 is 5. So the red is the random superposition. That's the actual observed solution. And you see that random and actual observed is very close. And since, yes?
What is the sequence identity?
On this axis is sequence identity, yes.
What does it mean?
So we take 2 3-dimensional objects, we superpose them optimally, and from that optimal superposition, I read off how many letters are identical on a string. So this is not based on a string alignment, it's based on a 3D alignment. And all I'm asking here really is, once I know their 3D structure, how similar would they be? So if the set of real structures would be like the red, then all structures would be related. There would be only one structure. Okay, they're not random. They differ. Structures differ. And the signal random in red and the signal all the structures here in black, the difference is this on average. The average difference is this. And that is essentially all you need in order to realize all the universal different structures. This is actually amazing. Now, so sort of the major point here is that down in this midnight zone, we do not have enough sequence signal. So even profile alignments cannot intrude. Most things that we actually possibly would want are down here and we never get them. That's why this is called the midnight zone. Next week, we will begin with structure prediction and in particular comparative modeling. For the time being, thanks for your attention, and I believe it's now the moment again that we will ask, that I wait for questions that will be sent.